Anyway, so today I'll be focusing most of the talk on Laurel Wilt, which is an emerging disease. We'll get through the whole story. The lab that I'm in, like Dave said, I work on wood decay fungi, but our lab, we do a lot of work with Laurel Wilt. We are intimately connected with this pathosystem. And so we'll tell you the whole story of that, and then I'll address some reasons why maybe emerging threats or exotics are becoming more and more of an issue, and talk about some that you might not be familiar with, or you are, and we'll just give you a nice recap. So it kind of all starts with this. We live in a world that's an international economy. Globalization is occurring. Trade is happening more and more often. And with this comes a lot of good things for the economy, obviously, because we can outsource things and get things made cheaper. The consumers are happy because they're getting cheaper products. But at the same time, from our background as foresters or forest health people, we have chances for introduction of pests. And so these things a lot of times can cause problems. And one of the reasons that these exotics cause a lot of problems for us occasionally is that the plants and the trees that we have here that are native to our region did not co-evolve with the pathogens or insect pests. So they don't know how to defend themselves, and a lot of times it results with epidemics, which we'll talk about some of those today. So with globalization, we get ships that come in with a lot of shipping containers, and in these shipping containers, you have a lot of solid wood packing material. And I'm sure you've heard this story before, but it's important to address it. And so with solid wood packing material, the reason that we use this is because it's cheap and it's affordable, and companies don't want to pay for plastic, which would be ideal for reducing the amount of pests, because a lot of pests come in on solid wood packing material, and we'll talk about some of those today. So yeah, a lot of those end up being beetles. There's a larvae from solid wood packing material there. And then once these things are introduced through these pathways, it's not just solid wood packing material too. I want to note that we're getting plants and plant products from other countries, and sometimes these are pathways that are getting introduced. But once they're here, we all know this story of moving firewood and moving plant material through our own nation. We are spreading the things ourselves. So continuing to campaign this message and let people understand how these things are getting here is important, especially through policymakers. And I'm not going to get into politics, but it's really important that we have funding for agencies that screen for these things. And funding is sadly not increasing today. So there was a study in 2014 where they looked at the amount of GDP, so how productive countries were, and each of these red dots represents a country. And they wanted to figure out if there was a correlation with, countries that are economically doing well and the number of forest pests. And there seemed to be, but there was two outliers, and those were UK and US. And they also looked at an analysis to look at amount of forested area. So if you're a country that has a lot of forest, do you have more pests? And there was really no correlation there. But once again, the US and the UK were outliers for reported number of pests versus these other parameters. So one reason that this could be is because the UK and the US share something in common, which is we import a lot of things. It's not the only reason that these things are outliers. Maybe we have better agencies that are detecting, we have better first detector programs and stuff like that, but importation is a problem. And so if you look at the amount of imports and exports over time in the US as a percent of the GDP, you can see that our economy has continued to grow over the last 100 years or so. But in the last 20 or 30 years, you can see a divide where we're getting more imports in as a percent of our GDP than exports. So we're importing a lot of goods. And that's good from an economic standpoint, but once again, these imports sometimes bring along friends that we don't necessarily like. So here's the solid wood packing material, the pallets that come in, and sometimes they're treated, sometimes they're not. It's hard to determine this, and policies should be in place to prevent this from happening, so we're not introducing accidental organisms into this country that can cause problems. So why is this all important, and bringing it out to a global scale of what this means? So this is a map of the world, and the colors correlate to the type of forests that are present in the various countries. And so you can see that at similar latitudes across the world, you have similar forests. But keep in mind here that China and a lot of Asia share very similar forest structure and composition to the eastern United States. So that's important, and we'll get to that in a second. One of the reasons it's important because in 2013, there was this huge project that was outlined, and they expanded the Panama Canal. So in doing so, ships can come into ports like New Orleans or Miami a lot quicker. So instead of stopping in California and then driving semi-trucks across the country, companies are now able to go direct via ship from Asia. And so why is that important? We go back to this map, and so the Panama Canal is here. So now things are leaving China, coming across, and then going through the Panama Canal and stopping in Florida. And so before, they would stop in California, and California, if you look at the forest structure, is not really similar to Asia. So a lot of things would get there, and maybe they didn't have a chance to create a founder event or an introduction that was successful, just because it's basically not a conducive environment for these organisms to thrive. Well, now they're going to be coming directly to the eastern United States, where the forest structure is very similar. We really share, it's kind of a mirror image of the types of trees that we have here. They also have in a lot of parts of Asia, and China specifically, because we import a lot of things from China. So this could be a huge deal. And so the Panama Canal expansion, they kind of campaigned with this: Florida, America's gateway to the world. And sure, it is. You know, we're going to get a lot of things more efficiently and stuff, and it can really boost the economy. But we like to think of it as Florida, America's gateway to the world's potential forest threats. And that's the truth of the matter, because with funding going away for regulatory agencies, people are still doing this, and they do have some increased port funding for APHIS and programs like that, but it's really not going to be enough. It's never enough. The things that are coming in are really hard to predict and really hard to notice, because a lot of times they're real small. And a lot of times in Asia and parts of the world where they're coming from, they're not really a problem. So we don't know that they're going to be a problem here. And that's how Laurel Wilt begins. So it all starts out in 2002 near Savannah, Georgia, in Port Wentworth. At a port, suspecting that it came in on a shipping container, they detected the beetle, which is the red bay ambrosia beetle. It's this little guy here. It's an ambrosia beetle, and just a little recap on ambrosia beetles' biology. These beetles, they don't feed on wood. So the typical ambrosia beetle, they carry fungi around to trees, and usually they go after, the typical textbook example is they're going after dead and dying trees. They're boring in, usually to the center of the tree, and they're depositing their fungus, and they're feeding on that. So they're some of the first farmers that we know about in biology. They're farming this fungus, and their young are living on it, and they establish communities, and then they fly off and establish elsewhere. This particular one here in the United States is a different story, and it's an atypical ambrosia beetle, and I'll get to that in a second. It has been traced back to Asia, to Taiwan, we suspect, is where the one that was introduced here came from. And in Taiwan and other parts of Asia, it's not a big problem. This beetle goes after dead and dying trees, aromatic family of plants, and it doesn't cause a whole lot of issues. So just keep that in mind. So since they detected it in 2002, they didn't think much of it. They identified it. They said, oh, this is whatever. It's not a big deal. Well, people started to notice in South Georgia and South Carolina that red bay trees were dying. And not just one here, onesies and twosies. It was lots of red bay trees were wilting and dying. So they didn't quite know what was going on. So you'd see forest settings like this, and Jekyll Island, and then you'd see things like this in forests where you just have these dead trees. And sure enough, they could associate the dead trees with exit holes from ambrosia beetles, and then they started to isolate a fungus out. And they had a big conference about it, and some people were debating that, maybe this is hydrology issues or other things. But you started to see some really good textbook symptoms of wilt disease. So these, usually you scrape the bark away and you have vascular discoloration. They were seeing that. They were seeing beetles, and they were isolating consistently a fungus. And so there's the beetle a little closer. And the fungus, it kind of grows as a yeast-like phase inside the beetle, and then it grows filamentous-like in the lab. And it produces these spores. So in this picture here, these little round ovals are all of the spores, or we call them conidia sometimes. But these are the propagules that are deposited in the tree, and this beetle feeds on it. And as it turns out, there was some genetic work done in our lab where we wanted to look at, we had isolates from all over the southeast, and we'll get to the distribution in a second. But we wanted to look at, are these things unique? Does this represent one introduction, or does it represent multiple introductions? Are these things potentially having sex and evolving here and causing a lot more problems? And it turns out that everything about this system is very atypical. The fungus is, all of the isolates that we have from the United States are of one genotype, and so they're clones. The beetles have a weird mating system as well. The males don't really live. So they're this system we call haplodiploid, and so essentially females have offspring, and it's essentially parthenogenesis, and these females that bring the fungus around, they're also clones. So we have a clonal beetle, one genotype, and you have a clonal fungus, and this is not common, and especially with the amount of damage that this thing is doing. So it's very interesting from a biological and academic standpoint. And it's also very interesting from a management standpoint because we have to learn about this thing a lot more than we know now, and we continue our. So the way that this pathosystem works is you have your beetles, they fly to a healthy red bay tree, and it's important to note that these red bay trees, they're aromatic. When you crush the leaves, you can smell them, and you have this kind of menthol-y type smell. They deposit their fungus, the plant reacts, you get wilt symptoms. Finally, the tree dies, more beetles come in, they raise their fungus, and then they go to a new tree. And so you get this vascular discoloration, which is typical of wilting diseases. So the lab that I'm in, we wanted to study how many spores does it take to kill a tree. And so Mark Hughes, who was a former PhD and postdoc in the lab, did exactly that, where he took known concentrations of spores of the fungus itself and then went and inoculated trees. And the way that he did this was he simulated beetle attacks. So at the very base of single trunk trees, he'd go in and he would drill with a small diameter drill bit, and then he would deposit spores at a known concentration, and then he'd follow it up again, because in nature, the beetles, it's not just one beetle that goes into the tree. It's one beetle comes in, and then the plant produces compounds, other beetles get attracted to it, another beetle comes in. So he would split the amount of spores into 2 inoculations, separated by 2 weeks. And then he measured disease over time. And so what's showing here is disease severity on the y-axis, and you got 2 different experiments. So the ones with the triangles are in the field setting, and the ones with the squares are a greenhouse. So there's a lot more control in the greenhouse. But what this is, the take-home message here is, this is 100 spores put into the tree. So 50 and 50 with the 2 inoculations. And just in a matter of like 40 days in the greenhouse, the trees were pretty much dead. You bump it up to 1,000 spores, and it gets a little sooner, and so on. So some other scientists, Tom Harrington and Steve Frederick, they wanted to look at, well, how many spores are the beetles carrying? One beetle, So to illustrate this, I told you that these beetles carry the fungus along with them, and they farm it. And they're usually selective for a preferential fungal species a lot of the times. And that's the case with the red bay ambrosia beetle. And so they have these fungal sacs, which are illustrated here, these little ovals that are called mycangia. And these sacs on the red bay ambrosia beetle are right behind the mouth part, so where that arrow is. And so they wanted to ask the question, in a red bay ambrosia beetle, how many spores are present in one? And so they went and collected beetles at a couple locations, and then they basically either squashed the whole beetle into some water and plated it out and counted how many colonies were on a plate, or they just took the mycangium, extracted it from the beetle, and then plated that out. And they found, on average, at one location, anywhere between 1 and 5,000 spores. But then at another location, they found 16,000 spores in individual beetle. And so if you remember from this, what I just said, trees can die in about 37 days with 100 spores. Well, they're finding a lot more than that. And they took it a step further and did some theoretical estimates of if the mycangia was at max capacity of spores, just estimating the volume of these fungal sacs and then estimating the size of the spores. If we packed them in there really tight, how many spores could they theoretically hold? And they found that these things could probably hold up to 30,000 spores. So this is a big deal. And the reason that it's a big deal is because, when the tree, when the tree dies, the reason that this happens, from what we're finding out, is that it's somewhat like other wilt diseases. It's somewhat of an allergic reaction. That's how I like to describe it, because the fungus and the trees have not co-evolved together. The tree has no idea how to react to it, but it knows that it's got this foreign invader and that it needs to do something. So whether it produces defense compounds or something else, well, in this particular case, like other wilt diseases, what happens is it recognizes the fungus that it's a foreign invader, and then it says, we got to keep this thing localized and contained. We got to keep it in one spot. And so it turns on some genes to produce these structures called tyloses, and these are produced in the xylem elements, which is the water-conducting tissue of the plant. And so in doing so, it does stop the fungus from moving through the transpiration stream and not moving in the plant, but it also stops the water from moving from the soil to the leaves, and you get the wilt symptom. So it's more of an overreaction of the plant than it is the fungus degrading the tissue itself. So that's an important thing to note as well. So moving along, it gets further complicated because although it was first detected on red bay and some of its relatives, swamp bay and silk bay, which are common along the east coast and the southeast coastal areas, they found out that this particular pathosystem, there's many plants that are susceptible to it, and they all happen to be in the family Lauraceae. So this includes, like I said, these close relatives, swamp bay, silk bay, but it includes avocado, which is a big deal because avocado is a billion-dollar industry, and people like to eat avocados. But it also includes sassafras, and that's where it's been detected here in Louisiana and in Arkansas recently. Laurel wilt attacking sassafras. And sassafras actually makes up a dominant forest species in the eastern United States. I don't think anybody's producing it for timber or whatever, but it does, it's important ecologically. But in addition to that, the list goes on. You've got California bay laurel. That's important if it gets out to California. And then Laurus nobilis, this is the one we cook with a lot of times, and the Mexican red bay. So it's made its way through all these hosts, and it seems that it can cause problems. And currently we're underway with a study that we started 2 weeks ago. We've collected seed and plants from collections across the world representing Lauraceae plants, and we're inoculating them and watching to see disease over time to see which ones have more of a risk than others. And we'll get to why that's important in a second. So since the initial introduction in 2002, when they first detected the beetle, these maps here are basically showing you the amount of percent mortality and then percent killed or wilt killed trees throughout the southeast. it's pretty much contained at high levels in the epicenter or wherever it was first established here. But you can see that it's spread all the way west to Texas, and it's also in southern Arkansas now too. But they've estimated that there's about 500 million trees that have died since the introduction. Think about that for a second. 500 million trees in like 15 years since they first detected the beetle. That's huge, and it's huge for a few reasons. Avocados obviously an economic problem, because if avocados start going down, people are complaining about avocado prices right now because of drought problems. We get a lot of avocados in this part of the world from Mexico and California, and they're having droughts, and so the prices are up. Well, think about if they start dying, how expensive avocados are going to be. And so we don't really want it to spread to Mexico or California. But culturally, these plants and the Lauraceae, since they have this aromatic scent to them, the American Indians have really prized this group of plants for a long time. And they think that it has these healing powers, and that it does a lot of good things. And whether it's true or not, they use it and they identify it as something that's important to their culture. So it's a really big deal. And in addition to that, you have biodiversity hotspots like the Everglades. And a biodiversity hotspot's an area that has plants that are endemic there and only there, and lots of them. And the Everglades is one of 5 or so in the United States. And the Everglades have these tree islands that contain mostly swamp bay. And these tree islands are being devastated not only by Laurel Wilt, but by invasive plant species, by invasive animals. You've heard about these giant pythons. There's a lot of problems down there, but Laurel Wilt is not helping. And finally, with ecosystems being devastated, you have rare and endangered species that rely on these types of plants going down as well. So this is a big problem. People say, I've talked to people about this before, and red bay, oh, who cares about red bay, But there's a lot of reasons to care about red bay, and hopefully by the end of this, you'll care about it as well. So here's a little GIF image that shows the spread since 2002. So you can see that it spreads from the epicenter in Georgia throughout Florida and across the southeastern panhandle there. But then, we need a little stationary one so we don't get a seizure. But then something weird happens. Like, what's happening here? How did this get here? How did it get to Texas? Any guesses? That's a good guess. Any others? Most likely wood, moving wood, exactly. So you've got dead trees. What do you want to do with it? You want to burn it. You want to go camping. You want to cut it down and move it across. And that's probably how it got to these areas. And like I said, in these areas, you're dealing more with sassafras because the red bay probably goes natural range is up the East Coast, all the way through Florida and across to East Texas. But this is a big problem because there's a lot of Lauraceae plants that people grow throughout the country. Not only the country, but the world. And so this map shows the concentration of forests that have Lauraceae as dominant plants in their ecosystems. And South America here, they have most of their dominant forest canopy is Lauraceae plants. And so it's really kind of scary to think that if this thing gets down there, they could lose ecosystems that are vitally important for biodiversity and ecological reasons. But in addition to that, you can see in the eastern United States, this is mostly due to sassafras. Sassafras is a dominant plant there. But like I told you before, where this disease or this organism comes from is China. And they have a lot of Lauraceae there too. But it turns out that none of their species are really susceptible to this. They don't have any disease over there, with the exception of one particular plant, and that plant is introduced to the country, and it's avocado. So recently they reported the disease on avocado, and but other than that, their Lauraceae are fine. So it's stuff maybe on the Western Hemisphere that we should be concerned about, and that's what we're doing this testing with currently. So yeah, we are campaigning at the University of Florida, and you can download this poster from the IFAS or the Extension website at University of Florida. We need to tell people about this disease. We need to tell them to stop moving firewood. It's the same old story with any invasive exotic. But just to show you the scale of avocado production and get into what's going on in avocado groves in Florida, we look at the production of avocado by unit of population density, and you can see that Mexico is, on this side of the world, the largest, the leading producer of avocado. And so since Laurel Wilt's been found in Texas, it seems like it's only a matter of time before it moves further south, and it's, the Mexican government, I think, is working with the USDA, and they're really trying to avoid this from happening because it could be bad. So that just illustrates it again. So in a avocado grove, this is in southern Florida, they were hit with Laurel Wilt, and they removed all the trees. That was the recommendation. It's like, you need to get rid of these things because they're acting as a reservoir for this fungus to be moved around by these beetles. But in addition to that, they found out that this thing was not just moving around by beetles. They found that it's similar to other wilt diseases like oak wilt, and the fact that it's actually can be transmitted through root grafts. And so you can see that these avocado trees get quite large, and they're planted pretty close together. And so it doesn't take a rocket scientist to figure out that could be a way that it can spread. And sure enough, it was. And so they're continually to see it down there, and they're managing it by removing trees and burning them. So here's another showing you a variety of the symptoms. So you can see this partial crown collapse or this wilt. These are some early symptoms of the wilting, because it looks like a lot like drought stress looks, because that's exactly what you're seeing. The xylem elements are getting clogged by these tyloses, which are kind of like balloons in the tissue. Water's not moving up. It kills it by drying out. So to make things even more complicated, they started to, when they were surveying in avocado fields for the red bay ambrosia beetle, which was probably how it got there initially, they were having a hard time finding it. And so you start scratching your head thinking, oh, well, how is this spreading? How is it getting around? And it turns out that they found 8 additional beetles that are relatives of Xyloborus glaberatus, which is the red bay ambrosia beetle, that could transfer this fungus. So some of them were carrying it in their mycangia, or those fungus sacs, but some of them were just kind of, it was an accidental introduction. They would have these spores that the fungus produced. Like I said, they kind of start off as a yeast phase. So they're really sticky, and they're large, and they can stick to the appendages of the beetles. And so they might be stuck to their mouth part, they might be stuck to their legs. They bore into the tree, deposit the fungus on accident, plants overreact, bam, dead. So the only one that's not shown here is there's a weevil species they found that also is accidentally introducing it. So that's kind of a big deal, and it has a lot of implications because, like I said, red bay ambrosia beetle, where it's been found so far is in the southeast, and sassafras, you're going further north across eastern United States, and maybe this red bay ambrosia beetle, we don't know, and maybe it doesn't survive the cold very well, and maybe that can keep it, knocked down in these areas that have a lot of sassafras. But if you have other potential beetles that are native here, or even not native, that are just cosmopolitan and they go into a lot of trees, it's possible that we'll have other beetles that can move it around for us, and even if the red bay ambrosia beetle doesn't die. So it's something to think about and also to continue to study. So I talked a little bit about the biodiversity hotspots, and these particular areas, there's 25 of them around the world. Most of them are in tropical or coastal areas, but if you look on the map, Florida, the Everglades, happens to be one of them. And so just a quote from a paper: the Florida Everglades contains one of the highest concentrations of species vulnerable to extinction in the United States. The 5,000 square kilometer wetland is home to at least 60 endangered species. So you've got a plant that probably a lot of organisms rely on that is being wiped out. So it's going to affect the ecosystem, and it's probably going to affect a lot of these endangered species as well. So this was some work done down in South Florida in the Everglades, where they were tracking Laurel Wilt percent coverage down there on their tree islands, and the blue dots are from 2011, and the orange dots are from 2013. So you can see that this in 2 years spreads very quickly, and so Swamp Bay down there are dying left and right. And to illustrate that, here's a helicopter shot, a view from a helicopter over the Everglades. All of those brown trees are Swamp Bay, and they're all dead from Laurel Wilt. So it's a big deal. It's really sad to go out in the woods and see this for most people. I mean, from a plant pathologist perspective, we I don't know, we get excited about this kind of stuff. But yeah, it's going to be bad for the ecosystem, definitely. And there's some work done in Mississippi where they were looking at a particular butterfly species, the Palamedes swallowtail butterfly, whose larvae particularly feed on Lauraceae plants, so these aromatic plants. And they looked at average number of butterflies caught in areas that were disease was not present, which are the white bars with the lines, or places where the disease was present, with the gray bars. And they found a significant difference, a reduction in the amount of butterflies they were finding in these areas. So these caterpillars weren't having the hosts to feed on and complete their life cycle. So it's contributing, Laurel Wilt is contributing to this decline. There might be other reasons as well, but Laurel Wilt is one of them. And what makes it even more important is this plant here. This is the orange-fringed orchid. And the orange-fringed orchid is pollinated specifically by this butterfly. And this particular orchid is threatened. So it's one of those species in an ecosystem that could be lost and go extinct because of Laurel Wilt, because of the loss of the Palamedes swallowtail. You get the loss of the orchid. That's the cascade event that could happen with something so devastating as this. So talking about the American Indians, we have some work going on with them. They're really interested in this, like I said, because this is a sacred plant to them. We have a campaign for them as well. They have different names for the plant, but one of the American Indian names is Tule, so it's not too late for the Tule. And so the American Indians are really interested from it from a medicinal and culinary standpoint. They use it often, using it in ceremonies and stuff. There's a whole slew of information about this that you can read about. But the important thing is they want to know how they can manage it so they can still harvest leaves. And so one of the things that our lab is doing is trying to figure out how we can do this for them. And so we're learning how to propagate them better, and we're doing some other management things that we'll get to in a second. But it's not all just American Indians. You know, we like to cook with Lauraceae plants or use them culinary as well. You know, sassafras tea, although it might be carcinogenic, once upon a time this was a popular thing to drink. And, you know, I'm from Louisiana, so you always find gumbo filet on the tables. Sassafras, that's what it is. Bay leaves and gumbo, it's a big thing. And so we use these plants as well. So they're maybe not economically important. People aren't growing them and making millions of dollars like avocado, but they are culturally important. And so what is being done in the lab that we're in and in other labs throughout the Southeast is we're looking at ways that we can manage it and also try to understand the biology better. So we had a project looking at fungicides. You know, other wilt diseases, you can inject trees with fungicides like Dutch elm disease. Preventatively, you can inject them with propiconazole and you get pretty good management out of it. And similar with oak wilt. So we wanted to test that with the red bay trees. In addition to that, we're learning how to propagate them so we can conserve these genetics, especially if we find tolerant species, which we'll get to. And also, we detected those other beetles, those other vectors that move this thing around, but is there other ways of spreading it that we're not thinking about because we're thinking of this pathosystem of this, tightly this relationship that's really tight between the beetle and the fungus, or beetles and the fungus. Maybe there's other ways of doing it too. So we've got some studies doing that. And finally, for the American Indians, this is the project, the coppice management study. So it turns out that the red bay ambrosia beetle is not really attracted to trees under about an inch in diameter, red bay trees or swamp bay. And so there's a graduate student in the lab who is doing a project where basically cutting the trees down and letting them regenerate to a size that the leaves can be harvested and used, and then doing it again. She's doing that as well as looking at fire as a potential management regime to, because it takes a lot of work to go in and cut all these trees and have them regenerate, so maybe fire can do it as well. And we're still gathering the results on that. But it seems like if the tree is not infected already, that these trees aren't being visited by the red bay ambrosia beetle. So that's good. And campaigning and outreach stuff is also important. We do a lot of that. That's why I'm here today. So this is Don Spence. He was Jason's, my professor's first PhD student, and he did a project where they looked at the fungicides. And long story short, there was a paper if you want to read the nitty-gritty details, but long story short is it works. This particular tree is a red bay tree. My office is right here in this building, and you don't find red bay trees like this in Florida anymore. They're few and far between. But this one's still alive. They've been injecting it for the past 7 years, once a year, with propiconazole at the labeled rate, and it works. However, there's a caveat there, and the caveat is this: the red bay trees are not like oak trees or elm trees in the way that they take up the chemical. There's a lot of gums produced in red bay wood. The wood's dense, and it can take a long time for the chemical to be taken up by the plant. And so it becomes a problem of realism. Is it realistic to do this on lots of red bay trees? Or should we focus just on featured trees like this? And so that's kind of, we need to figure out maybe more efficient ways to get the chemical into the plant so it can do its stuff. But the chemical itself does work. And we have another project going on with some fungicides in the lab now for that, and hopefully we'll have data soon. So as far as the propagation study goes, Mark Hughes, again, he did all this work actually. That's him there. He's now in Hawaii. Poor guy. He's working on a wilt disease there, Ohia wilt, which is destroying their forest as well. I'm trying to figure out that. But anyways, so when he went out and collected and did survey work of diseased plants, one of the things that he noticed was there were these specimen trees that were large and seemed to not be affected. I mean, an area where everything else was dead, there'd be this one large red bay tree. And so, before we can think about what's going on and why this is happening like this, it's like, well, let's get some seed and let's get some cuttings and figure out how to grow this thing, because there's something to these plants, we're calling them survivor trees, that make them resistant. So here's a paper from Native Plants Journal where basically laying out the best, most efficient ways to propagate them, so we know how to do that now well. And so just grow them up in mist benches and then pot them up. You get them into small saplings, and then we planted them out in the field at one of our research stations. And so we really wanted to determine, are these things truly resistant or tolerant to this disease? And so the way that we do this is we inoculate them artificially. And so we use the same protocol where we go in, these are single trunk trees, and we do 2 sets of inoculations with 1,500 spores per mil per each tree with 2 applications like I talked about earlier, and then monitor disease over time. And so some of these trees in this picture are checks. We always have to have checks to make sure that the disease is going to work and our spores were alive and happy and everything. But as it turns out, the 2 trees on the top here are both quote-unquote survivor trees. So you might be scratching your head, well, what's going on? Why is the one on the right where he's standing, why is that one dead? Well, come to find out, we think that there might be 2 types of tolerance or 2 types of resistance. One towards the fungal side, where the fungus gets in and the plant's not overreacting as much. And then one towards the beetle side. Maybe these plants are producing chemicals that are repellents of the beetle, and so the beetles are just grazing over them and they're not visiting them and depositing their fungus. So we wanted to look at that a little further. So we identified some clones. We're currently looking at genes that are coding for this resistance. We've done some work looking at the xylem elements and looking at particular structures to see if that's part of the resistance, and then also the beetles. So just to show you, so this is some electron microscope of some wood cuttings, some slivers of wood, and these large circles are the xylem, or the water-conducting tissues. And on the left, you have a tree that was inoculated, and this was 30 days after inoculation, so it was dead. It was susceptible red bay. And on the right, you've got the tolerant red bay. And so there's a clear difference between them. The water-conducting tissues of the susceptible, they have those balloon-like structures, that's tyloses present, and the tolerant one has minimal amounts of tyloses being produced. So it's tolerant because it's not overreacting like the majority of tree species. So that's pretty cool. And to show the same thing at a different longitudinal angle, the xylem elements are these pipes here, and on the left you've got the susceptible one, and these structures, these balloon-like structures, are the tyloses, and on the right you see none. So that's pretty cool. We figured out a mechanism for this, and potentially if we can figure out what the genes are, we can create breeding programs that really go after that and really kind of tune in on, let's not have these plants overreact. And this is an issue that is common with these epidemic type situations. People want to learn how to manage it. You know, the first question is, oh, what can I spray for it? I mean, everybody knows that. What can I do to? But resistance always comes way later. And you know, I think that's an important thing that we're doing, looking for why are these things resistant, so we can get it before it gets really bad. So we have these plants that are these tolerant ones to the fungus, and we're currently doing some conservation work at our biological station at Ordway Swisher in Florida, where we have these plants planted out. And so it's kind of like the beta test, where they're in an area where Laurel Wilt is, and we're going to watch them over time, and at the very least we're regenerating that forest that was lost with these quote-unquote resistant ones. And in addition to that, Jason's been contacting multiple public gardens, botanical gardens around the country, and he's depositing these accessions there. So we have a germplasm for these tolerant plants, not just in University of Florida, but elsewhere as well, in case things go wrong. So going back to our planting out here and the other type of resistance, so the beetles. So we wanted to determine, are there volatiles that are repelling beetles? And so far we haven't looked at the resistance versus susceptible, but we just kind of wanted to see over the course of, after we inoculate susceptible plants, are these plants, are they attracting beetles? Are there chemicals being produced, defense chemicals, that are going to repel it? And so this is the infection, you know, at day 0, plant's healthy. By 3 days, it's still asymptomatic. At 10 days, you start to see some chlorosis and early wilt symptoms, and then by 20 days, the plant's dead. So what was done by some people in the lab was they collected volatiles, so these aromatic chemicals that are being released as gases from across time of this disease system. And they collected the volatiles, and then they did this nice insect behavior assay where they had this 4-way olfactometer where they had plants that were control plants, they weren't inoculated, and they collected the volatiles from those across those time points, and then they had plants, or the volatiles collected from plants that were inoculated over those time points, and then looked for, they put the beetles in the middle, right about here, and they let them decide. So the way the volatiles would come in and they would recirculate through these areas. So 2 of them would have the inoculated plants, and then 2 ways would have the control plants. And the beetles got to make their own decision. Where do I want to go? Do I want to go towards the volatiles of the inoculated or towards the control? Okay. So what they found was that at day 1 day after inoculation, there was really no difference. There was no selection. Some of them went to the control, some of them went to the inoculated. But at 3 days, it changed. There was something in the volatile profile of these plants that were inoculated that was deterring the beetles. So the beetles chose significantly more to go to the plants that were not inoculated. So there's something there, but it totally changes again at 10 days. So once the plant starts to show symptoms and die, the beetles are actually favoring those trees, which makes a lot of sense, with the biology of ambrosia beetles in general. So they took it a step further and wanted to look at what are these chemicals at the 3 days, the time when they were deterring the beetles from going there, and they found two that were only produced at large concentrations at the 3 days. One of them was DMNT. I won't attempt to pronounce the chemical name. And the other one was methyl salicylate, which you might be more familiar with. This is the minty smelling stuff that's in your toothpaste, and it's widely used in a lot of processes. But it's also a precursor to a lot of defense compounds in plants. So it makes a lot of sense that this will be produced after the inoculation, because these chemicals are along pathways that help the plant defend itself. So what one of Mark's studies, what he wanted to do was see, with mountain pine beetle out west, they're using this splat stuff, which is like a silicon-based material, and they're mixing it with verbenone and putting it on trees, and the beetles seem to not visit these trees because it's repelling them from them. So if this chemical methyl salicylate is a repellent, can we use it for the Laurel Wilt pathosystem to protect these trees? And so we picked an arbitrary concentration of the chemical. We got splat made for it, so it slowly releases this chemical out. And he took recently felled red bay bolts and put them on these poles, and put the splat on the log, and then put a sticky card underneath, and then would count beetles every week. How many red bay ambrosia beetles are coming to the logs that have methyl salicylate? He also included verbenone and some combination. So you've got methyl salicylate and verbenone, methyl salicylate by itself, a non-host poplar at one location, and we used laurel oak at another, and then a control, just red bay. And it turns out that pretty much all of the treatments were better than the control. So if you were just a red bay log with nothing, essentially, you find more beetles at those logs. So that's pretty cool. And methyl salicylate by itself, it didn't do as good as the verbenone plus methyl salicylate or the verbenone alone. And why this is interesting and important to us is verbenone's expensive. So it might work and stuff, but maybe it's not a cost-effective strategy. But methyl salicylate is very cheap. And so we're working out right now with different concentrations of methyl salicylate, could you have a better effect of repelling these beetles? And to take this a step further as well, it wasn't just sticky traps. We're not just counting beetles out of sticky traps, but he took these bolts back to the lab, debarked them, and then actually counted exit holes. So how many beetles are coming in? And it turns out the red bay ambrosia beetle hole is the same diameter as a paperclip. So it was nice, it was the easy way to count them. Just put all the paper clips in and count all the paper clips that were in the tree. And we got similar results there. So the positive control, the blue bars are one location and the orange bars are another. There were differences in the amount of beetles at the two locations, but positive control for the most part was a lot more than the negative control. So our controls work. That's always a project in science. You got to get those controls to work. And the chemicals did similar things. So we saw a reduction, but the verbenone and the methyl salicylate plus verbenone seemed to be a little bit better at controlling these beetles. So like I said, we're doing this again at higher concentrations, and not only that, we're not just using logs in the woods, we're actually going and finding trees that have survived that aren't infected and doing it on living trees to see if it could work. Because the idea here is if we take tolerant species and we plant them out as a conservation effort, maybe if we couple that with applications of this, maybe we can bottleneck this beetle and fungus and it'll be displaced and we won't see it anymore. That's the hopeful idea. So another complicated issue that we're learning about now. I took this picture is from 2 weeks ago. There's a new postdoc in the lab who's really studying this from a molecular standpoint. I don't understand half of what she says. But one thing I do understand, which is really interesting, is that she can isolate the fungus not only from the wood of infected trees, but from the leaves. And so this gets a little more complicated. So she inoculated these trees, and then from leaves of the same tree and bark about 30 centimeters or so from the inoculation site, she isolated from both the leaves and the bark of these infected trees. And after 6 days, she gets similar amounts of colonies from the leaves that she does from the bark. So remember, we had those 8 other beetle vectors, and if we keep thinking about this from a ambrosia beetle and fungus relationship, we might miss stuff. There's a lot of other insects, especially insects that feed on the leaves, maybe some root weevils or plant hoppers and things like this that potentially can move from plant to plant and spread this thing too. So we're working on that as well to find additional vectors. This could be some bad things, and not only that, but pruning and shearing plants is a thing that happens in avocado groves. And so this could be another way that if we are pruning things and we prune the leaves or the tissues that aren't infected and move it around, this could be something that could be bad. So to be continued on that. So since it's been introduced, it's spread far and wide. It's attacking a lot of the members of Lauraceae. We're looking at other plant species that it is going to attack. We have found some resistance, and we're working with that, like I said. We're looking at other means of dispersal, and we're continually trying to figure out new and innovative management ideas. So I'm going to switch gears a little bit and talk about some other emerging things that are coming up that you might be familiar with somewhat, and other ones you might not be. So other exotic diseases that were introduced to the United States that are of interest. Laurel Wilt, obviously, we talked about that. Thousands cankers. This one was introduced to the eastern part of the United States from the western part. Also a beetle and a fungus relationship. And from what I can tell, it's kind of, it's very, people that are studying this, they're having a lot of problems finding the beetle in the walnuts. So, it might have been tightly related with weather patterns and drought. They talked about drought, and that could have been why we saw those walnuts die. But it's something to still continue to think about because we don't want to forget about it. Date palm lethal decline. A lot of people from maybe the more middle part of the country won't have this because you don't have palm trees. But in South Texas and southern Louisiana, especially in Florida, there's this big problem right now that's killing a lot of palm trees. And specifically, it's killing our native palms, the sable species. And we'll talk about that. And sudden oak death we know about, and we'll talk a little bit of that. But the Dutch elm disease, white pine blister rust, and chestnut blight that were introduced, some of them hundreds of years ago, they're still causing problems today. I mean, it's pretty much impossible to find American chestnut. You can find a few of them here and there. But this disease, the fungus, Cryphonectria parasitica, it likes to hang out as a saprophyte on a lot of our native oak species. So the fungus isn't going away. It's got a place to live, and if chestnuts can't make it. But they are doing some great work with breeding. That's a whole nother talk. So then there's also insects. So this disease, this date palm lethal decline. So I guess I have somewhat of an intimate relationship with it because after I finished my master's at NC State, I was a county agent doing consumer horticulture down in the New Orleans metro area, and I rode around with this arborist down there several times. He's like, "Man, there's something going on with these palms. They're just dying." And these were old growth palms, and they were melting. And I'll show you the symptoms in a second. And so we went around, we looked at them several times. We sent some samples to the diagnostic lab, got really no results that were meaningful because these plants kept on dying. So we reached out to Texas A&M, who had dealt with this disease, date palm lethal decline, also known as Texas Phoenix palm decline. But the people in Texas said, no, we're not calling it that. We're not having another Texas root rot thing on our hands. So they're trying to change it to this. But anyways, so they confirmed that this was the particular disease that was killing these trees, and it's caused by a phytoplasma, which is basically a amorphous bacterium. It has no cell wall or structure. It's just a bunch of blobs, and it lives in the phloem. They don't really know the vector of it yet, but other phytoplasma diseases seem to be transmitted by leafhoppers or plant hoppers. They're working on. They actually hired 2 extension faculty or researchers to study the vector of this in Florida recently. So they're putting a lot of money and effort. So it was first identified in Texas. They described symptoms of this in Texas, in the southern Texas area from the 70s, but they didn't know what it was. And then in 2002, they identified it. And then it was introduced to Florida, they think, in 2006. So that's when they first found it. And it's attacking a suite of palms. So there's another phytoplasma disease on palm called palm yellowing, or lethal yellowing, which specifically goes after coconuts. But this one goes after a lot of palms. So Canary Island date palms, edible date palms, these are not natives, but cabbage palms. And I don't know if they found it on Sable mexicana or Sable texensis in Texas yet, but definitely is affecting Sable palmetto, which is a big staple. I mean, it's the state tree of Florida. So there's still not much known about this. But this is a situation that I took this picture in New Orleans, and so you can see that these trees, they end up just having this melted canopy here. So it kind of goes, there's a suite of symptoms. You got this yellowing, and then you've got browning, and then eventually come to that. So this is date palm lethal decline. So in Texas, 2002 they found it, 2006 in the Tampa area, and then 2013 is when we found it in Louisiana. And it's probably been here a lot longer than this. We just didn't know what it was because you can't. That's the other thing is you can't really grow this organism out in the lab. You have to do it by DNA detection, and so that is very costly. And a lot of times people don't want to pay the money. But this disease is probably more widespread than we know about. So again, symptoms can have this chlorosis, almost look like some nutrient deficiency, and a lot of times they are nutrient deficient as well. But one of the things is this canopy gets like this, it collapses. I like to call it melts, Here it is on Sable palm, a similar thing. It first has this bronzing appearance. The spear leaf always dies. That's an initial symptom. And then the whole canopy can actually fall out of the tree, and you get this decapitated palm trees. And a bad joke, but ISIS is not taking credit for these decapitations. So anyways, the diagnostic symptoms are this yellowing from the bottom up and then followed by browning. The spear leaf dies, the fruit drop early, the flowers will die, and this crown melting thing. So in this picture, spear leaf's dead, you get this bronzing and the premature fruit fall. And show you that from this picture. So you still have flower stalks on this Canary Island date palm, but you got the yellowing. And then further along, these trees have no flowers because they're dead. And then you get the spear leaf's dead in this plant. Actually, the spear leaf was dead in all these. I went up in a bucket truck and I could just pull the spear leaf out with no, which if you ever tried that on a palm tree, it's really fibrous tissues, and it's really hard to do that if the tree was healthy. And finally, you get that dead tree. So this is a emerging thing that's becoming more and more of an issue in palm growing areas. And it's something to be on the lookout for if you have palms. So there are management tactics. If you find you get a positive diagnosis, usually removal is the recommendation because it can act as a reservoir because insects are most likely moving this thing around. If the spear leaf is not dead, then you can inject with a bactericide, oxytetracycline, at the labeled rate. And in palms, you don't have to put multiple injection sites. You can just do one. And you got to check, labels in your area for what's available because a lot of these are just labels that are specific to certain states because it's not so widespread or they don't think it is. So you always need a sample. I mean, if you suspect this, it's a good idea to get in contact with your extension people, Texas A&M, Louisiana, and wherever else you are if you have palms, and so on. So the other one that will probably be a refresher for you because you've heard about this is sudden oak death. Millions of oak trees have died from this disease. It's caused by a Phytophthora species, causes cankers on trees. And the disease cycle is very interesting because it can kill mature oak trees in a matter of 3 years. And they did some tests early on, showing that this Phytophthora can cause a canker, and by itself alone, it might take a little bit longer to kill the tree. But what happens is you get a canker, the plants stress, and they get mass attacked by beetles. And then so in 3 years, 100-year-old oak trees die. And the way that this thing is surviving around, you might think, oh, it should have killed off all the oak trees in California and Oregon. But it has reservoir hosts, actually a Lauraceae, California bay laurel is one of them, and also some other plants that they don't really die from it. They get some dieback symptoms, and they can eventually die. But they can harbor this fungus or this fungal-like organism for a long time, and then it can spread that way. They've also found that it can move in waterways very efficiently, which is unique for Phytophthora groups because these organisms, we don't know them as saprophytes really, but they can travel like at least a kilometer in a stream of water. So it's kind of important from that standpoint. So here's a setting in California with a bunch of dead oak trees. They get these cankers, they bleed or gum up, and you get this bleeding effect. And then where the canker is, with Phytophthora cankers, you always see this black line perimeter. So if you ever have a Phytophthora canker problem and you scratch away at the canker in the transition zone, you'll find this black line that's pretty diagnostic. So since the original detection in the early 2000s, it really hasn't moved from a standpoint of natural spread. It's pretty much in California and southern Oregon, Curry County, Oregon. When this is interesting, so I'll get to that in a second. So then this is the amount of bay laurels that have died or have infections over the course of weather patterns. So you're looking at dry, once this goes back to dry conditions and wet conditions, and it looks like once you have dry conditions for long enough, which they've been having a lot of, the infection or the disease incidence goes down. So that's kind of interesting. And then you have a wet year and it picks back up, and then it goes down after dry years. And so there's the spread from 2014 more recently. But in 2008, they came up with this. This was a risk model of predicting where Phytophthora ramorum would thrive. And it turns out it really hasn't gotten to these natural areas, which is good. It's good news. But the Appalachian region was one of the more higher risk areas just based on the composition of trees that they have there. Lots of oaks, lots of understory Ericaceae plants that can be these alternate hosts. And that's Phytophthora ramorum has a host range of hundreds and hundreds of trees. And so if it does get into these natural areas, it can be bad. So we still need to be on the lookout for it, but it doesn't seem like it's spreading naturally. There was a spell, it's federally quarantined, so it is important to be on the lookout for it and contact the right people if you do think you find it. But it, I've lost my train of thought there. But the thing is, it hasn't gotten there. Oh, I was going to say that there was a spell in the 2006, 2007 where nursery moved a bunch of plants around, and you can still detect the organism in a lot of these locations throughout, even in the East Coast today. And you can still go out and find this fungus, but it hasn't escaped into the natural world. So it is something to keep an eye out for. We have a suite of oak trees that are susceptible and would be bad if it got into the natural stands. But don't be fooled. There are a lot of other Phytophthora species, some of which aren't nearly as aggressive, but can cause problems, and they look somewhat similar. So you get these bleeding cankers. You do get the black lines around the canker. This is Phytophthora cinnamomi, which is pretty widespread in the south. So keep in mind there's other Phytophthora species, not just because you have a Phytophthora canker that it's ramorum. So I won't go too much. I was going to talk about emerald ash borer and the Dutch elm disease, but we've covered that, so that's good. But yeah, we didn't learn from our mistakes, especially in urban areas. We planted ash trees in a lot of these areas that were wiped out. But anyways, we'll move on to the replacement. Okay. So emerald ash borer is a problem. I'm not skipping over it because it's not a problem, but it is. I do want to mention this. There's other bark beetles or wood-boring insects that attack ash and that are native. The ash bark beetle can cause dieback symptoms, and stressed ash trees almost always have this beetle there. And so, we need to teach people that there's other beetles too, but we need to be on the lookout for both and track this as it spreads. And it's probably in areas that we don't know at this point yet. So Okay, don't move firewood. So I'll tell you about a program through the USDA, APHIS, and CFIST, the CAPS program, or the Cooperative Agriculture Pest Survey and Pest Detection. They have a nice website with a lot of good information on future threats. So you can peruse this website at your leisure. I'll talk about one of them in a second, but in oak tree they have this list of potential pests, and if they were introduced to this country, it would be bad news. One of which that's interesting to me, and I'm really personally interested in it, is Japanese oak wilt. It's also caused by a Rafflesia species, the causal agent of Laurel wilt. This is Rafflesia cursivora. And currently it's been found just in Japan up to this point. And it first detected in the early 2000s, but it's been spreading and killing. It's killed over 200,000 oak trees in Japan. And so it's caused the beetle. There is a beetle that spreads it. It's a Platypodon beetle, and it doesn't have those specialized fungal sacs, but it has these weird pits. Just a trivial fact there, and the fungus lives in those little pits there. But the fungus is a Rafflesia, so it looks similar. It has these really sticky conidia, and it can move from oak tree by these beetle spreads, and it causes wilt symptoms. So same thing. The plant's overreacting. It's clogging up its tissues, and the plants are dying from the drought stress. So if this gets to this country, we have a lot of oak species, and a lot of these oak species are susceptible to this, and so we really need to be on the lookout for this and educate people about Japanese oak wilt. So Japan's over here. It has, once again, similar forest composition. It's always a good idea to learn about other forests and where we're importing goods from, and to teach people about the importance of this. Because if you have similar species, then these similar pests can come over here and cause problems by accident. So it all goes back to this globalization and our international economy of how we get things introduced. Whether it's plant material, our solid wood packing material, we're bringing these things here ourselves. And we're not, but other people are, and people that don't really, they're not tree people. We're all tree people, so we understand this stuff a little better, but we have to educate and continue to talk to people who are tree ignorant and teach them about the implications of this international economy and moving things around. So this is just a map. This was a paper where they looked at insect pests and diseases that came in that were introduced in the late 90s, and then again in the late 2000s. And the numbers of introduced organisms just exponentially increases. So as our economy gets better, our pests are becoming more prevalent, and things are getting introduced, and they're causing a lot of problems. So in Florida alone, if you look at where these pests are coming from, this is between 86 and 2000. Most of our pests are coming from Asia and then the neotropics, so in South America and the Caribbean area. And so if you think about the goods that are coming from these two locations, most of the stuff coming from Asia is commodities that's probably video games or toys and stuff that's being produced over there, coming in probably the solid wood packing material route. Whereas stuff from neotropic is more plant material, more plant-related stuff. And so we're getting things that way. And similarly, if you look at it just across insect orders, you can see Coleoptera here. So between the 70s and 80s are the red lines, and the green are more recent. And you can see beetles are coleopterans are dominant things that's being introduced. But in addition to that, some sucking insects as well, and some butterflies, caterpillars. And so most of these are unintentional introductions. But I do want to say that once upon a time, when we didn't think the way we think now, lots of people wanted to introduce things because they were used to them in Europe or wherever they came from. And there was actually, I read about this recently, there is a society called the American Acclimatization Society, and they were based out of New York. And it's one of the reasons that we have the European starling here today, and the house sparrow, and some of these other things that are invasive, because people wanted it to be like home. So they brought things here from Europe, introduced them, and now they're invasive. I mean, there's 3 birds that are not protected in the United States, and it's the pigeon, it's the European starling, and the house sparrow. And all of those were introduced. The pigeon introduced by the French in the 1600s, and the other ones more in the 1900s or so. But regardless, we used to introduce things intentionally. Now, we don't think like that anymore. We're introducing things by accident. And some of these things, like the Laurel Wilt system, we don't even know they're going to be a problem. So it's this cryptic pathosystem introduction. So, but keep in mind that there's a lot of things that we find that are exotic, that are introduced, but they don't really cause a whole lot of problems. The majority of them. So the blue line here is the insects that are coming in that don't really cause problems, and these are the high-impact insects. So there's very few that are actually being able to establish and cause problems. But these few are causing lots of problems. So if you look over across feeding guilds, the majority of these are foliage feeders and sap feeders. But if you look at it across which ones are causing the most problems, it's the wood-boring insects, which mostly are beetles, but some wasps as well. So this is a big economic problem that continues to needs addressing and educating. So there's several stages in which we can slow these invasions. We can prevent them through regulatory ways, policies, lobbying for more funding for the federal government and the Forest Service and USDA APHIS. We can also work on things like when they're establishing to try to control them if we find that they're here, as well as what we're doing like with the Laurel Wilt, working with resistance and management tactics. So it's going to be costly to do a lot of this stuff. Monitoring efforts will cost money, and the budget doesn't seem to be going that direction. In addition to that, if we say solid wood packing material is against the law, and we make companies not use that stuff, and they use something like plastic, it's going to cost them a lot of money, and in the end, it's going to cost consumers a lot of money, and people aren't going to be happy. So there's a lot of innovative ideas that we need to be talking to economists and business people to figure out a solution, because this is a problem. So I'll leave you with this picture of this ship, because there's probably some introduced insects that are in there. And actually, one more picture that I'm happy that I got to come back to Louisiana. I don't get to come here very often, but I did put in, that's what I ate last night over at Crawdaddy's, and it was pretty good. So thanks for having me here, and if you have any questions, I'll be happy to take them, or we can talk at lunch. Thank you. No, that's a good question. Yeah, probably more than likely they're going to die. If they don't have something to grow on a substrate, they're going to die. They're not like other fungi that produce these survival structures and persist like Phytophthora morum does. So they're not going to survive very long, but they are very good at living in dead woody tissue. But there's this, like forests have succession, you cut down a forest, you get grasses and then conifers and then eventually hardwoods. There's also succession with dead and dying trees and the fungi that they invite in. So once a tree is dead, it might be that the Raphael loricula might get displaced by something else that's saprophytic. And we're seeing that there's actually it's just an interesting side note, there's a wood decay fungus, this Fomes fasciatus, that seems to be highly correlated with Laurel Wilt killed red bays and decaying it. So we are seeing that, but yeah, it's probably not going to survive in leaves that have fallen, and probably not for long after the tree is decaying as well. So it doesn't do anything other than carry it in this sterilized sac. So the fungus does live in there, and beetles, they regulate temperature well in there, and so this fungus actually lives as this yeast-like phase inside the beetle. And it turns out that the fungus is really hard to find in these dead and dying trees. It's not establishing like other fungi. Canker fungi, though, you can actually see mycelium grow, and they're releasing enzymes to degrade the tissue and stuff. This particular one's not the case. We rarely see mycelium at all in trees. It's these conidia, these little spores that are microscopic that are causing the tree to overreact and kill itself. So it doesn't take much. And I think that's the atypical story of laurel wilt is it's a very low amount of fungus that's needed to kill these trees. And it's really the trees killing themselves. Right, and that's a good idea. There's always a little hesitation with introducing other things into the environment, obviously. But we are currently, the postdoc, June Lee, this lady who just came on board who's doing this fungus-host interaction study where she's doing these really fancy techniques where she's looking at gene expression in the plants over periods of time after the plants have been inoculated to really target which genes in the plant are causing the excessive tylosis production. And then the idea is that we can target these genes in breeding programs. But in addition to that, we are also looking at the fact that the genes that might be affecting, on the fungus side, the genes that are causing the plant to overreact, we're looking at it from that standpoint too. And we do have a close relative. And currently, we also have, I didn't mention this, but in fungi, sex is really weird. It's not like animals, so these fungi in their native range, they have two different mating types. So you can have a genotype here, that's one mating type, and for it to actually produce sex and exchange DNA to evolve over time, they have to meet in nature and then produce sexual structures. So far, they haven't found the sexual stage of this fungus in nature, but currently I'm working with an undergraduate student right now where we have mating types from Asia, and we're doing this all in a contained lab, so not outside at all. But we're partnering up the two different mating types to try to force sexual reproduction. And so far we haven't been successful. But I'm thinking about turning up that Barry White music, and get the magic happening. But yeah, so we're looking at it from that standpoint. We haven't really talked about actually releasing these things. It hasn't gotten that far as a biocontrol deal, but we are trying to target it so we can understand it better. Yeah, so the question is, how is it going to affect sassafras in the future, and are there differences between the two hosts? So they're both aromatic species. They both have odors and volatiles that are attractive to these beetles. And in places where sassafras is being hit, it's being hit pretty hard. I actually haven't seen it in nature on sassafras. If anybody has any locations nearby, that this is happening, because it is occurring in northern Louisiana, I would like to see that. But yeah, it's destroying them. So the real question isn't how is it going to affect them. We know that it can kill these sassafras trees. The real question is how is it going to move into our more dominant sassafras forests, and will the disease act the same way there? And we think that it will, because it's also an overreaction is what we think with sassafras as well, and all of the susceptible species we found thus far. So it's a matter of getting the fungus there, and that's to be found out, I guess.
Yep.